Hi, my name's Eden Houske, and I'll be presenting on the role of the primary cilium of renal epithelial cells in fluid flow mechanotransduction. So cellular mechanotransduction is the transmittance of a mechanical stimulus into a biochemical response. It plays a vital role in cell function and in the development and maintenance of tissues and organs. Renal epithelial cells are subject to shear stress from filtrate, which is detected by a structure called the primary cilium. These cilia recognize the stress and transmit it into a response that initiates various metabolic pathways. When the cilium is absent or defective, regulation of cellular mechanotransduction is lost or impaired, which leads to detrimental effects such as renal cyst formation in autosomal polycystic kidney disease, or ADPKD. ADPKD is a hereditary disease that results from mutations in PKD genes, which these code for transmembrane proteins called polycystins. Polycystins aid in mechanotransduction, so when they're mutated, the result is a dysfunctional primary cilium. Loss of polycystins, such as PC1 and PC2, causes a decrease in calcium influx, which results in fluid secretion. This ultimately causes renal failure due to the development of renal cysts in the kidneys from this fluid secretion. Currently, there are no cures for ADPKD. The most common treatments include either dialysis or kidney transplants. Both of these treatments wait until the kidney nearly fails, and they've also proved to be painful and problematic for patients. It's important to determine the role of the primary cilium in fluid flow mechanotransduction so that we can potentially work towards pharmaceutical treatments aimed at halting metabolic dysfunction in the kidney. So, although certain pathways have been associated with PKD, there's currently not a clear understanding of the mechanisms through which cellular mechanotransduction is initiated by the primary cilium and how this process exactly operates in the kidney. The field is also lacking information about which metabolites are correlated with mechanotransduction. So our first goal of this project was to expand our understanding of fluid flow mechanotransduction in renal epithelial cells. It's recognized that the primary cilium does play a vital role in cellular mechanotransduction, but not much is known about how exactly the cilium participates in this process and what happens exactly when the cilium is defective, such as in individuals with ADPKD. This leads us to our second goal, which was to determine how mechanotransduction is altered when the primary cilium is removed. This would essentially allow us to gain insight into dysregulated mechanotransduction in PKD. So we hypothesized that cells lacking primary cilia would have a greater evidence of dysregulated metabolic pathways compared to cells with intact cilia. We tested this hypothesis by emulating the disease model of mechanotransduction seen in ADPKD, and then we ultimately analyzed the results by comparing cells with and without cilia in both static and fluid flow stress environments. So for our methods, Madin-Carby Canine Kidney cells, or MDCK cells, were cultured using a growth media called DMEM. And so we passaged the cells multiple times prior to carrying out the experiment in order to prevent overconfluency. And the cells were then resuspended at a concentration of 300,000 cells per 2 milliliters for plating in the 6-well plate, and then they were incubated for 24 hours. So we essentially had two groups. One had cilia and the other did not. And within each group, some were exposed to shear stress and others were exposed to a static environment. Ammonium sulfate was added at a concentration of 30 millimolars to 3 wells in each plate to remove the cilia. And old media was then replaced with experimental media, which essentially inhibited cell division during the experiment. So one 6-well plate containing cells with and without cilia was placed on a platform rocker at a 6-degree angle at 0.74 hertz for 5 hours. This was designed to simulate fluid flow stress, and the other 6-well plate was placed in a static environment. So to extract our metabolites, we submerged the cells in liquid nitrogen in order to break them open so that we could gain access to all of the metabolites. Cellular debris was removed through centrifugation, and we mixed the metabolites with 80% volume-to-volume methanol to extract them, and then mixed them with aqueous acetonitrile to get rid of all of the undesired proteins. We dried down the metabolite solution in a vacuum concentrator to remove solvent, and then used liquid chromatography mass spectrometry, or LCMS, to elute the metabolites over time depending on their polar properties. Compounds were essentially ionized and passed through a mass analyzer, which allowed us to identify all of the different metabolites. We used a global approach to identify all of these metabolites, which would allow us to identify significantly affected metabolites that could be further investigated in future experiments. We used a variety of univariate and multivariate statistical tests to assign metabolites to different cohorts. Another program called MSPK pathways allowed us to detect specific pathways that were altered directly as a result of cilia removal. So looking at the ANOVA in figure 2, we could see there were 2002 metabolites detected across all cohorts, with 3 metabolites, which are shown in red, being significantly different from one another. Table 1 just simply shows numerical values for those 3 significant metabolites. So both of these figures show hierarchical cluster analysis visualized as a dendrogram and a heat map. These tests were used to visualize metabolomic profiles, identify subgroups of sample sets, and to determine if cohorts were distinct from one another. Separation or clustering of samples was clearly visualized within each cohort. And so 2 groups were detected, including one that had cilia and the other that did not. This is shown by the separation of the 2 major branches of the HCA, which is the figure on the left. There's one irregular occurrence, which is the presence of a ciliated group in a non-ciliated branch and vice versa. But ultimately, the data suggests that metabolic pathways were mainly perturbed in response to the absence of cilia rather than in response to fluid flow exposure. So we used principal component analysis to visualize any variation between the 2 cohorts. There ended up being a slight separation detected between group C, which was fluid flow with cilia, and group D, which was fluid flow without cilia. So this basically tells us that the removal of the primary cilium does indeed alter metabolic pathways. Supervised clusters and underlying variation between cohorts was detected using partial least squares discriminant analysis, or PLSDA. There was a large amount of overlap between the fluid flow and the static cohorts with cilia, as seen in purple and green, and there was little overlap between the fluid flow cohort without cilia, which is seen in blue, and any of the others because this was the diseased model. There was also some overlap between the static with and without cilia cohorts, which is seen in green and red. PLSDA identified specific groups of metabolites that contributed the most to separation between cohorts. So metabolites with VIP scores that were greater than 2 were mapped to metabolic pathways using MSPK pathways. And ultimately, from this, we can conclude that since there is some separation between cohorts, we do have a shift in metabolism in the different conditions. So table 2 shows that the only significantly perturbed pathway was the ether lipid metabolism, because it was the only group with a p-value that was less than 0.05. Other pathways that had high numbers of metabolites detected included tryptophan metabolism, fatty acid elongation and degradation, and biosynthesis of unsaturated fatty acids. So ultimately, we were able to separate cohorts of metabolites, and we identified ether lipid metabolism as being a perturbed pathway related to altered mechanotransduction. This makes sense because renal cysts develop as a result of widespread cell proliferation, and a greater number of cells is correlated with a greater number of phospholipid bilayers. So essentially, we can conclude that ether lipid metabolism is altered in PKD, which causes the formation of these renal cysts. Also, many of the other insignificant pathways we identified included those involved with fatty acids, such as fatty acid elongation, degradation, and biosynthesis of unsaturated fatty acids. So if this experiment was repeated with a higher cell confluency, some of these pathways may turn out to be significant after all. So we can conclude that global metabolomes were primarily perturbed based on the presence or absence of the primary cilia. Due to the irregular distribution of the dendrogram, we have to take into consideration the role of the cilia removing agent in this study, which was ammonium sulfate. Ammonium sulfate may have had significant effects on some of the molecules and ultimately interfered with the clustering of our metabolites. Some limitations of our study included failure to visualize cilia removal using immunofluorescence, less than 70 to 90% cell confluency, and our sample size was quite small. Immunofluorescence was unsuccessful due to the use of old 4% paraformaldehyde, which essentially failed to fix our cells, and instead these cells were washed away, which explains the lack of visualization of fluorescence under the epifluorescent microscope. This visualization was ultimately very essential to confirm that the cilia were indeed removed. Also, the low confluency of the cells could provide a reason behind the low metabolite abundances and the insignificantly affected pathways. Our study was only conducted once for each environment, which accounted for a small sample size, so it would be beneficial to repeat this global metabolic profiling with more replicates to ensure we would get similar results. In future studies, a targeted metabolics approach would be beneficial to focus on specific metabolites involved in ether lipid metabolism. And so ultimately, the ether lipid metabolism pathway should be further investigated to clarify how exactly it contributes to the cause of PKD. I would like to thank Dr. Hahn for her guidance throughout this project, as well as the other biomechanics students who participated in this research with myself this past fall semester. Thank you.